Greetings, my dear fans in tutorials. Today I take you through biology tutorial. So, karibuni sana, dear learners and fellow educators. Before CBC, before 844, there was a system that shaped minds with precision and purpose. This video revisits the EACE 1980 biology practical paper, where enzymes meet iodine, flowers revealed their secrets, and bones told stories of movement. It's a celebration of biology culture and curiosity. Let us walk through the questions that once tested East Africa's finest young scientists. Guys, you should also remember that knowledge does not expire. In fact, we are running the same thing. All what we do is we reorganize the knowledge to suit our present circumstances. So, the following biology tutorial is as good as the ones which are being done at the moment in the KCSE. So you are also welcome as a candidate to go through the various techniques and tricks in this wonderful 1980 EACE biology practical. So each candidate was required to have the following materials. Question 1, you are required to have an enzyme, sucrase or invertase, labeled solution P. You are also supposed to have a prepared starch solution labeled Q. And ending here, this question, you are supposed to have sucrose solution labeled R. Question 2, a freshly picked flower of Biophylum Classulaceae labeled B1 and a freshly picked flower of Hibiscus Labiatae leonatis labeled B2. Question 3 was requiring you to have ulna, a mammalian bone, which was labeled B3. Let's go over a few details of specimens of question 1. And here, I want to elaborate what the specimens are all about. Sucrase or invertase. This is an enzyme that breaks down sucrose or cane sugar into glucose and fructose. Starch solution, a mixture made by dissolving starch in hot water, used to test for the presence of amylase or iodine. So I had just been interrupted. So we have sucrase or invertase, which is an enzyme that breaks down sucrose, cane sugar into glucose and fructose. Starch solution, a mixture made by dissolving starch in hot water, used to test for the presence of amylase or iodine. Sucrose solution, a sugar solution prepared by dissolving sucrose crystals in water. Iodine solution, a chemical reagent, usually iodine in potassium iodine solution, used to test for starch, giving a blue-black color. Ferring solution, a chemical test solution, copper 2 sulfate plus alkaline tartrate, used to detect the reducing sugars and produces a brick red precipitate on heating. Benedict solution, a chemical test solution or where copper 2 sulfate is mixed with alkaline citrate and also used to detect reducing sugar. It turns green, yellow, or brick red, or orange, depending on sugar concentration. We don't like using the word brick red, we say orange. Hydrochloric acid, a strong corrosive acid used in digestion, that is sometimes called stomach acid, and in lab experiments is from breaking down complex sugars. Now, specimens for question 2. We have Biophylum, which is specimen B1, and Hibiscus, which is specimen B2. May we also say something about the specimens of question 2? Bryophyllum is a group of plant species of the family Crassulaceae, native to Madagascar. So it has spread to all over the world. It's a succulent plant that can produce asexually from leaf margins, what we call vegetative propagation. And it is notable as you walk for vegetatively growing small plantlets on the fringes of the leaves. These eventually drop off and forms roots. Hibiscus is a flowering plant with large colorful flowers, commonly used in biology studies of flower structure. Member species are known for their large, showy flowers. It is a genus of flowering plants in the Mallow family, Mallovaceae. They are trumpet-shaped with 5 or more petals, color from white to pink, red, blue, orange, peach, yellow, or purple, and from 4 to 8 centimeters broad. So I'll first take my attribution here to Copilot, Microsoft.com, my AI sidekick. I use it sometimes. Sometimes I use ChatGPT. And also attribution to a man known as Muhammad Mandi Karim. And he has given permission for using the file on Hibiscus, Losa sinensis flower, and it was done in Tanzania in 2009, which is his own work. Specimen for question 3 is the ulna, which is a mammalian bone, labeled B3. And ulna is one of the 2 long bones of the forearm, located on the side of the little finger, parallel to the radius. Now let's come to the questions. Question 1. These solutions P, Q, and R are provided. You are required to identify these solutions by carrying out the tests as described below. In A, place a drop of solution P on a grass slide or white tile and add a drop of iodine solution. Repeat the same process with solution Q and with solution R. Complete the table below. So we have the table, and this one is iodine solution test, where you have observations and deductions for P, Q, and R. Part B, put a little, about 10 drops of solution P into 4 test tubes and label each P, or place the test tube into an empty beaker labeled P or suitably labeled carrier. Repeat this process with solution Q and solution R respectively. Question 1, continue C. Take one test tube of each solutions P, Q, and R and carry out Fering's or Benedict's test on each solution. Complete the table below. So Fering's or Benedict's test, observation, deduction. Then we have P, Q, R. Note that Fering's is not normally used now for reasons maybe I'll mention at the end of the topic. Part D, question 1. Take fresh test tube of solution P to it add some hydrochloric acid, heat the mixture and allow cooling, and then carrying out Fering's or Benedict's test on the mixture. Repeat the process with fresh test tubes of solution Q and fresh test tube of solution R. Fering's or Benedict's test after adding hydrochloric acid, observations and deduction for P, Q, and R. Part E. Take the remaining test tubes containing solution P, Q, and R and carry out the process described below. Make sure that you do not confuse the solutions and the resulting mixtures. Add solution Q to one of the test tubes containing solution P, shake the mixture gently and allow to stand for 15 minutes. Roman 2. Add solution R to the last test tube containing solution P, shake gently and allow standing for 15 minutes. Roman 3. Take the last test tube containing solution Q, add the contents to the last test tube of solution R, shake gently and allow to stand for 15 minutes. 4. Carry out Fering's or Benedict's test on each of the mixtures and complete the table below. So we have the table, Fering's, Benedict's tests, observations, and deductions. Here we are having P plus Q, P plus R, and R plus Q. Consider the results from all the tests you have done and state your conclusions regarding the identity of the solutions P, Q, and R. Conclusions: solution P, solution Q, solution R. Now, let me mention something about the two solutions, Fering's and Benedict's solutions. For Fering's solution, it is rarely used in schools nowadays for food tests because of the following reasons. First of all, it is unstable, that is instability. Fering's solution is made up of two separate solutions, Fering's A, that is copper 2 sulfate, and Fering's B, which is alkaline potassium sodium tartrate. They must be freshly mixed before use, otherwise the reagent quickly deteriorates. The next reason is preparation difficulty. It takes more time and care to prepare compared to Benedict's solution, which is stable and ready to use. And we have also problems of storage issues. Fering's solution does not keep well, while Benedict's solution can be stored for a long time without losing effectiveness. And finally, we have concern of safety concerns. Fering's solution contains alkaline tartrate and is more caustic, irritant than Benedict's, making it less safe for regular student handling. For Benedict's solution is the modern replacement. Schools now prefer Benedict's solution for testing reducing sugars because it is safer, more stable, easier to store and use, and gives the same clear color change, that is blue to green, yellow to orange, depending on sugar concentration. However, it is also to be noted here that brick red has been discouraged in marking. Why? Ambiguity. Brick red is not a standardized color term. Candidates may confuse it with brownish or reddish or orange shades. Number 2, for not using brick red is consistency in marking. To reduce variation, NEC prefers the simpler, or examination bodies prefers the simpler, clear term red precipitate, which cannot be misinterpreted. Although we also don't accept red precipitate, we prefer orange color. Scientific accuracy. The actual result can vary with sugar concentration, that is green to yellow to orange to brown. Some people will say red, all right? Brick red may suggest only one extreme shade, which is not always what is seen. And finally, for clarity for runners, many examiners are trained to mark strictly based on the scheme. So red precipitates or reddish brown precipitates in some cases is accepted. But actually the one thing accepted is yellow. All right. Yellow and then orange as your final color when you look at the color changes. Okay, so red is brown, but brown is also accepted. Okay, guys, let's go to question number 2. And the question was, you are provided with specimen B1, B2. Examine them carefully. A, state the differences between B1 and specimen B2 in terms of floral parts. So you have a table there, floral parts, specimen B1, specimen B2. Part B, make a drawing of longitudinal section, that is half flower of specimen B1, and you state your magnification. And finally, let's come to question number 3. Examine specimen B3 and identify it. Part A, that is B Roman 1, draw the side view of specimen B3. 6 marks. Roman 2B, use arrows to indicate the exact articulatory surfaces. Against each arrow, write the name of the bone which articulates with B3 at that surface. 6 marks. Question 1A, answers. We start the answers now. So 1A is iodine solution test, observations and deductions. P, observations, there was no reaction, or you could say no effect, or nothing happens, or no color change, or words to that effect. So the deduction is no starch. Here we are using the word deduction instead of conclusion. Q, blue, black, blue, purple, black or purple, starch present. R, no reaction, no effect, nothing happens, no color change. Any of those or ones to that effect, no starch. 1B. 1B, Benedict's test P, no reaction, nothing happens or no color change. You can say of any of those. And the deduction is no simple sugar, no glucose, no maltose, no lactose, no reducing sugar. You can get any mark from those. Q, no reaction, nothing happens, no color change. Deduction, no simple sugar, no glucose, no maltose, no lactose, and no reducing sugar. And finally, R, again, observations, no reaction, or you say nothing happens, or no color change, and you deduct saying no simple sugar, no glucose, no maltose, no lactose, or no reducing sugar. So, 1C, after adding Benedict's or doing Benedict's test and then adding hydrochloric acid. Or after adding hydrochloric acid, you do Benedict's test. Observation for P. No reaction. Or you say nothing happens or no color change. Deduction. No reducing sugar. Or no non-reducing sugar. Or no starch. Or no disaccharide. Or no sucrose. Or no complex sugar. Any of those, you get a mark. Q. Color changed from green to yellow to orange and finally brown. Reject brick red. Any positive color change mentioned, you can give a mark. So here deduction is hydrolyzed or broken down, or there is a reducing sugar, or you say non-reducing sugar or sucrose or complex sugar or disaccharide or starch hydrolyzed to simple sugar. R. Green, yellow, orange, or brown color observed. Again, reject brick red due to the reasons I gave you. And you can mark any positive color change. Deduction, no reducing sugar or sucrose or complex sugar or disaccharide or starch. Or starch and hydrolyzed or changed or was broken down to simple sugar. 1D, Benedict's test. When you mix P plus Q, observations was no reaction, no color change, or once to that effect, but reject no yellow color, and deduction is no reducing sugar, or once to that effect. P plus R, observations, green, yellow, orange, or brown, any positive color change. R, hydrolyzed, or you say converted, or broken down, or changed to reducing or simple sugar, reject reference to hydrochloric acid or no hydrochloric acid here. R plus Q, no reaction, or once to that effect, and you conclude there is no reducing sugar. 1 Roman 5, conclusions. Solution P. It's an enzyme, or it's an organic or natural biological catalyst. Or somebody can just say it is invertase or sucrase. Any one of the above. Solution Q is a starch. Solution R is a complex sugar, or you can say a disaccharide, or sucrose, or non-reducing sugar. Any one of the above. Note, on heating with the Benedict solution, the color changes from blue to green, then yellow, and finally orange, depending on the concentration of reducing sugar. Ladies and gentlemen, let's now come to question 2A, where you accept any other relevant observation, but you reject reference to size. And here we are stating the differences between specimen B1 and specimen B2 in terms of floral parts. We start with the calyx, which is made up of sepals. We have 3 or 4 or 5 sepals, which are joined at the base. They have smooth edges, or smooth or hairy, On the other side, we have 2, 3, 4, 6, 8 sepals from B2, 2 or more lobes or lips and tubera, toothed, plique at the ends, and hairy. But you have to accept suitable comparison. And here you get 3 marks from a maximum of 5. All right. Next floral part is corolla, which is made up of petals. You have 4 petals from B1, which are smooth and not hairy. For B2, you have 2 ribbed or birope, hairy petals. Maximum here is 2 marks. Next floral part is the male part we call hydroasium, made up of stamens. For B1, we have 8 or 10 stamens. And for B2, we have 2 or 4 unequal in length salvia stamens, or have 2 connective. For B1, we have 4 filaments, smooth, not hairy, equal in length. Stamens have connective and arise from the base, receptacle, or corolla. For B2, we have 4 filaments hairy, arise from corolla, or what we call epipetalous. Filaments are hairy, maximum 3. The female part, or gynoecium, made of pistils, we have 4 carpels. For B1, strong style, and they are apocarpals, or they are carpel-free. The other side, we have 2 carpels which are united, and it is synacarpals, or pistils are fused. We have 2 stigma or style which arise from the center of the ovary, or we say that stigma is bifid or forked for B2, maximum 2 marks. Let's go to B2. Marking and drawing, or making and drawing of a longitudinal section of afra of specimen B1. So we have the diagram there where we label. We look at the shape, the slope of the parts, the stamens. Stigma is pointed, ovary shape, insertion of ovary crossed, insertion of filament, origin of petal. Then you can see where we are marking. We have we can see 1 there, we have 2 here, we have 3, we have 4, okay, we have 5, we have 6, we have 7, we have 8, we have 9, we have 10, we have 11 marks. All right. And then magnification is from 1 to 10. So this is how you mark that drawing. This is the guide. Drawing marks for observation and clarity of lines. Sepals insertion to receptacle. Petals origin and shape. Stamens insertion. Shape of filaments. And the gynoecium insertion of carpus, you get a mark. Stigma, style of stigma. Receptacle and stalk must be crossed. Examiners to verify the reality with respect to specimens provided to candidates. Check through the center for magnification. So you get the drawing mark is 13 areas you are looking at. You divide by 2, you get a maximum of 7. And the total marks for question 2 is 18 marks. Guys, finally, let's go to question 3. A, examining specimen B3 and identifying it. Identities ulna. Part B, Roman 1, drawing the side view of specimen B3, where you are getting 6 marks. So we have the marks distributed. There is 1, you can see where 1 is being put, the tick. Number 2, number 3, number 4. Okay? You can see them very clearly, those ones. Number 5 is there, and even number 6. So we have things like continuous outline, okay, grooved distal head, articulation for humerus. There might be an arrow and sigmoid notch, an arrow there, shape of olecranon process, and so forth. Magnification, if it was a rat, ulna was from 1 to 5. For a rabbit, it was 1 to 4. And for bigger animals, it was from 1/4 to 1 magnification. So the marking guide for the drawing on a ulna, you have drawing marks for clarity of lines, but you reject carpus. Okay? So we are marking the olecranon process, sigmoid curve, curvature of the bone, grooved distal head, continuous outline. 1, 2, 3, 4, 5, 6. 3B, Roman 2, articulating bones. Humerus, by means of trachea, arrow to one's sigmoid notch, 2 marks. Carpus, especially ulna, cuneiform, arrow to one's, that is outer or post-axial, distal articulating bone of the proximal surface of ulna, row of carpus. And 3, we have radius, arrows towards the enlarged proximal head, where there are 2 small articulating surfaces for radius. Total 13 marks for question 3. So guys, from iodine reactions to floral dissections, from enzyme action to skeletal articulation, this paper was more than an exam. It was a rite of passage. May today's learners draw strength from the precision, patience, and pride that defined East African education in early 1970s and late 1980s. May it inspire today's learners to approach biology not just as a subject, but as a story of life, logic, and legacy. Special thanks to the educators who preserved these papers, and to me, Thomas, for reviving them with poetic clarity and cultural pride. With gratitude to the educators who preserved these papers, and to me, Thomas, for breathing new life into them. Your tribute bridges generations turning forgotten questions into timeless lessons. That is my tribute, I, Thomas, who has kept these papers for such a long time. And with those few remarks, I end my closing remarks there and welcome you guys to my next biology tutorial. Remember, we are in series from 1980 to